another video to show you today. Okay, so back here. Substitutions are just going to be a single nucleotide change. So instead of saying ATC, what would normally base pair with this for DNA? What would normally base pair? TAG, right? So instead, maybe DNA polymerase makes a mistake. Instead of putting an A there, they put a C there. So that's just a substitution. It doesn't shift it left or right. But if you instead, we had that same T, and you inserted an A, or you took out the A there, you would have a shift one way or the other due to the deletion or insertion called a frame shift mutation. All right, mutations are mostly beneficial and useful for organisms. True, false? Mostly thumbs down, yeah. Okay, so mainly false are mainly, they're neutral, so they're silent mutations, or they're usually detrimental. There are a couple of positive mutations where it yields something that's more beneficial for that environment, specifically wherever that organism's living. Because like antibiotic resistance, they get a mutation, what happens to the bacteria? If they get a positive mutation, what could happen to the bacteria in the presence of an antibiotic? They could become resistant to the antibiotic and live and make more babies. The goal of life, right? So they could definitely do that. All right, mutations can only occur during interphase. When is what are the parts of interphase? As a quick review. G1, S, and G2. Right. Okay. So which of these is a part that's we've been focusing on? S. We've been focused on S, which is synthesis, which is DNA replication. All right. So mutations can only occur during that. Is that true or false? False. When else can it occur? Mitosis. Yeah. So when an anaphase doesn't split correctly, so you get a non-disjunction happening. What about like when you're making that mRNA? What's that process called of making mRNA? Transcription. You can have a mistake in transcription, which would then yield a poor amino acid that would not be the right one for the protein made. So that's false. Not all genes are turned on or activated. True, false? True, right. Because, for example, what kind of cells would photoreceptor genes be turned on for? So plant cells, what about in the human body? Photoreceptor genes. In the eye, photo, light, receptors, something that recepts or takes in light. You probably wouldn't have photoreceptor genes turned on in your bladder, right? Like, there's no reason for that. That would be a waste of resources to turn on those genes and express them, like doing transcription and translation. That would just be a waste of amino acids. Digestive enzymes, what cells would you need those genes turned on for? Stomach cells. Would you want it in your bone cells? No, because then it would digest your bones and you'd be like that man. You know what I'm talking about. This one? What's the dance? No bones? Yeah, there you go. Okay, here. True. Mutations can be genetically inherited? True. Like, yeah, we knew that. Okay. I'm not going to go through the last ones. We already know that. All right. So I have a video for you for mutations in another video, and then we will start presentations quickly. Oops. All right. Quick video. We kick the lights for me, Brandon. Okie dokie. This is 1 minute. You got this.
A point mutation is a change in the nucleotide sequence of a gene. This can result from an error during replication that is not corrected by the usual repair mechanisms. Under normal circumstances, adenines and thymines should bind to one another, as should cytosines and guanines. In a base substitution mutation, a non-complementary nucleotide is incorporated into the new strand. The resulting altered triplet may code for a different amino acid, or even a stop codon. Here we see 4 codons and their corresponding amino acids. A change within the second triplet results in a change in the amino acid. None of the other amino acids are affected. Insertions and deletions result in frame shift mutations. The insertion of an extra nucleotide not only affects the triplet it is part of, but all subsequent triplets, thus drastically altering the protein product. The result is similar, if a nucleotide is deleted.
Oh no, frame shift. Okay. I also have, I know, you guys always look at what's afterwards. You're weird. Okay, so I was a doofus and videoed upright, and we all know that's a no-no, right? You have to video sideways on your phones. Yeah, didn't think about it because I was trying to get the second video started. So I had to flip it on YouTube, which it did not like. And the quality's a little poor, so deal with it.
You are not in the nucleus, right? They're in the nucleus.
It's
And you guys put down your other colored paper. Keep only the blue for now. You'll pick it up in a second. Okay, and DNA double helix, go.
You guys are very excited.
Okay, DNA unzip. mRNA, go.
I was a busy body, don't everywhere we went there.
mRNA transcription. Okay, mRNA link together. mRNA link together. mRNA leave the nucleus. mRNA leave the nucleus. DNA drop, yes. Okay, mRNA is now in the ribosome. Okay, transfer RNA, please find your amino acid. Find your amino acid. Oh, I'm the first. Hope it's wrong. Nope, just the first. Okay, actually here, Sean, be start. Okay, and translation is going to begin with the start codon. Start codon, go. Go, go, go. Start codon. Start codon. Okay, hold it up. Here we go. tRNA uno. Tell us what it is. Gag. Base pair, yes. Okay, amino acid next. Yes, bond to the amino acid. Oh, look at it. They didn't get the walk in, man. All right, work it, people. Work it. Work it. Oh yeah, they're really getting it. Woo! Working it. Look at this huge amino acid chain. There you go. There you go. We brought that amino acid left. And we have the stop codon. Okay, amino acids together. 3D shape. Go. Lovely. Okay. We now disassemble. Interpretive dance. And then everybody bow. Thank you. Thank you very much. Pick up your pieces. Let's go.
Yay! And it was less than 3 minutes. You're awesome people. Okay, so where are we at? Bio. Okay. Pretty much I have to show you one more thing, and then I'm gonna let you guys present. What's my last thing? Oh no. Oh yeah, here it is. Okay. On your sheet or on your desk there should be a white sheet of paper. If you don't have one on your desk, share with the person next to you. What I need you to do is read, listen from your past. You're going to read your article. This is based on the discoveries of biotechnology. What two fields do you think biotechnology pulls together? And technology. So it's tools to discover more about in-depth knowledge about biology, about your body, about DNA, about proteins, about things to fix the ailments of the human body. You realize that, oh, this was just the other day, my husband sent me this. In mice, they found a way to stop cell aging, and they have started human trials. So I'm expecting to live, I'm thinking at least 120, 125. It depends on how well I take care of my body, but you know, we're going to live longer and longer and longer. So they have to start getting technologies to, you know, to help that process. And they're going to come up with other things because, you know, we're going to need different things in 100 years. Okay, so other tools are coming out of the woodwork to help with the longevity of the human species. FYI, North Carolina is number 3 in the nation for biotechnology companies and jobs and discoveries and patents on technologies and methods. North Carolina is number 3 out of 50 people. We're pretty good. So this is the place to be if you're interested in biology, you're interested in biotechnology or biomedical research. This is a big thing for us. So if you like anything we're talking about, think about it as a career. FYI. Okay, so I'm going to read this too. You guys are going to pick out 3 fun, interesting things that you read about, and you're going to be sharing with them with somebody. So write down your 3 interesting facts, okay? So take about 3 minutes, okay? Who does not have one? Yeah, write on your own sheet of paper. Who does not have one?
What is it?
I'll tell you about that. Actually, come in. You guys are sharing one. and I don't mind doing it. Write down your 3 fun facts or interesting facts on your own paper because you're going to be walking around and sharing in a minute. All right, I will start. Mine was most awkward, so I'm going to start with it, okay? And this is on video. We're going to talk about erectile dysfunction, okay? So, fun fact. Men between 40 and 70 years of age, 50% of them have erectile dysfunction. Guys, you got something to look forward to. Okay, so the problem is that there's a gene that's in the DNA, it's coding for a protein. Sorry, I tried to straight. Okay, it's not funny. Okay, so there's a gene that controls the contraction of soft tissue. You can imagine what that means. So if the soft tissue, or if the gene is not going to be active, then therefore that person has erectile dysfunction. There's also other components to it: overactive bladder disease, irritable bowel syndrome, and asthma. That was the most interesting one, though. Okay, so what they can do is take gene therapy, put a good gene in where that gene is not functioning correctly, and therefore cure this problem. Okay, so that is something that's happening now. This was in 2006, 8 years ago. Guarantee you they've made progress on this. So you guys are going to go around and share 1 to 2 of your facts that you found. Give a little bit of a briefing of your article. Take about 5 minutes. Hit up as many people as you can, okay? Write down 1 of their facts on your paper. Write down 1 of their facts. Take about 5 minutes. Get up.
I know his mom died. I must keep it there. Drop high school. His freshman year. One way to be younger is to reduce his calories and that's basically my theory on there's ways to catch on light and reduce oh, disaster! That's so unfortunate. I know, right? Did you hear that? Yeah.
Did you share it, Joey?
Here's all three of them.
Oh, okay. Find one more person. You have 1 more minute. Ten seconds.
I can still do stuff for you.